now. Okay. So while if you have to ask any questions, please raise your hands before asking any such question. So if you raise your hand, I'll be getting a notification and I can then stop and maybe let you ask the question. You can even use the chat for that purpose. Now coming to the next slide, what is aging? Aging is a time-dependent process where there is a progressive loss of physiological integrity which accumulate over time. So it is not it is a process that happens with time, and there is a loss of integrity, uh, very similar to what happens in case of cancer, where there is accumulation of mutation. Aging is believed as of now that it cannot be reversed. Uh, Aging leads to impaired functions, increased vulnerability to death and diseases. And if we know the mechanism of aging, we can devise some pharmacological intervention to delay this process, right? So aging, it plays a very important role in case of cancer, like in case of if you see prostate cancer or even breast cancer that happen in older men and women, right? Diabetes, cardiovascular diseases, this happen in older people. And then we have neurodegenerative disease. We will be talking about these diseases like Parkinson's and Alzheimer's, which happen at a later age. Now, coming to the differences between cancer and aging. So these two, just a minute, just give me a moment. Okay, so there is a difference. Essentially, aging, in case of aging, there is a progressive loss of function of cells and tissues, right? But in case of cancer, the cells undergo uncontrolled growth. There is uncontrolled proliferation in case of cancer. In aging, there is a reduction in regenerative ability of the cells. So the regenerative ability means that when there is loss of cells, when there is death of cells, or where it could be due to injury or it could be due to normal apoptosis, new cells, they come in and replace this void. So that is the ability of regeneration. Aged cells, they do not have this property of regeneration. Just a minute. Okay. In case of cancer, there is no regard to the natural process of cell death. There is no regard to apoptosis. So cancer cells are known to evade apoptosis. In aging, there is an apparent loss of fitness of the cell, and in cancer, there is an apparent gain in fitness. So gain in fitness in this is, what does that mean? It means that cancer cells sometimes are better suited with the environment and they grow more in number than in normal cells, right? So gain in fitness does not necessarily mean good to you, but it means good survivability of the cell. So it's I'll upload the lectures, yes. So aging and cancer, they seem to be, opposing forces. There are still people joining in. Okay, they seem to be opposing forces. So there's one force of aging, there's another force of cancer. So we will see how these two things are related with each other. So let's go to the next slide. Now, what happens is, in case of aging, cellular damages that could be due to mutation, there could be other sort of damages, we'll talk about it. They accumulate over a long period of time, and it essentially prevents the cell to grow any further. Fine. But however, in case of cancer, usually what we see that some cellular damages could also provide a growth advantage to the cells. So, for example, if there is a mutation in a tumor suppressor gene, then uh, you know what is a tumor suppressor gene and what is an oncogene? Anybody would like to say something on this? Okay, this is an interactive course. I would expect somebody to answer, or I have to take names.
No, sir. We're not aware of the terminology.
Okay.
Sir,
Hello.
sir, can I say?
Yes.
So can
Sir, oncogenes are the genes which are responsible for cancer, or it causes some. If there is any kind of mutations here, it causes cancer. And
Right.
another one that you said that please can you say the term? I'm not getting it.
Tumor suppressor gene. Are you not able to see the like slide? It's here.
Sir, actually my network problem. Anyways,
Okay. Okay.
tumor suppressor genes, it does not allow the cell to grow, or it suppresses it, as far as I know.
Kind of. So you're right, actually. So oncogenes, they cause cancer, tumor suppressor gene prevent cancer. That's the very easiest terminology. We'll talk about it in the next slide only. So let's talk about one of the diseases, a coronary artery blockage, which is called arteriosclerosis. As you can see that in the early ages, there is almost no blockage. But later there is lipid accumulation in the artery and the artery starts getting blocked. Then there is aneurysm or rupture of the blood vessels. And also there is thrombosis, which happens at a later stage. So there is a progressive, accumulation of lipids or a progressive, accumulation of this damage that leads to these kind of diseases. Even the diseases that is caused due to aging, that happens due to this phenomenon. Now, age, hence, it plays a very crucial role. So we'll talk about it. Now, coming to the next slide, just a minute. Okay, so cancer research, there were fundamental discoveries which were made when cancer was discovered. People started studying about cancer. The first being of tumor suppressor gene and oncogene. So as one of you already mentioned, that tumor suppressor gene prevent cancer, oncogenes promote cancer. So that's the simplest definition. And then the role of tumor microenvironment was also discovered. Like when cancer is formed, when there are cancerous cells together in a vicinity, the microenvironment or the environment of those cells, they change because they go through a different sort of metabolism. So let's say the pH of these cells, they decrease, or these cells undergo hypoxic growth because of the lack of oxygen. So many things happen. There is also mechanistic differences. So like cancer cells, they are different mechanically than normal cells because they can grow beyond their capacity, beyond the capacity of a normal cell. So hence we can say that cancer has multiple layers of complexity. It is not a very simple process. It's also not a single hit process. It's not like you get some mutation and the other day you have cancer. It's not like that. Rather, these mutations, they start accumulating. A lot of mutations happen over time, and then at a later stage, you get some cancer. Now, to understand cancer, there were two important research articles that had come up. Uh, both from the same group, Hanan and Weinberg. This is the name of the authors, basically. So they discovered 6 hallmarks of cancer, and later in 2011, they discovered 4 additional hallmarks. So what do I mean by hallmark? So for an event to become an hallmark, it has to manifest itself in the development of cancer. It's if you increase experimentally this event or this characteristic, then it should promote cancer, it should accelerate the development of cancer. If you reduce this event, if you reduce this characteristic, it should prevent cancer. So that's the idea. That's what hallmark mean. So these are the properties required for an event to qualify as a hallmark. So what are the hallmarks? So in the left hand side, you can see the first 6 hallmarks. So definitely evading apoptosis. That means cancer cells, they do not undergo normal cell death. Programmed cell death. That is quite clear. then self-sufficiency in growth signals and insensitivity to anti-growth signals. So usually normal cells, they are dependent on mitogenic growth factors. You know, these are extracellular factors that signal a cell to grow. But cancer cells have developed mechanisms that they have their own cell growth machinery. They do not require any other stimulus. So how do they do that? We'll see that. And also they're insensitive to anti-growth signals. So basically in your system has positive loop, feedback and negative feedback. If there is too much of growth, there will be a negative feedback. There will be anti-growth signals. But cancer cells, they are immune to these anti-growth signals. Then there is tissue invasion and metastasis. So essentially, cancer cells, they are not limited to their own, tissue or the same organ. They can travel to other tissues and organ. So that happens due to metastasis. So that process is called metastasis, and it involves like endothelial to mesenchymal transition, and then there is angiogenesis. So angiogenesis is the development of blood vessels. Okay, so these blood vessels are very important so that these cancer cells, they can get nutrition, because it is also very important. So through these blood vessels only, these cancer cells, they travel to different other parts of the body. Another important hallmark is a limitless replicative potential. So here in this course, we will be discussing that normal eukaryotic cell have a limited replication numbers which they can go through. After that, the cell cannot replicate further. We will see how that happens. But in case of cancer cells, they have limitless replicative potential. They can replicate lifelong, I mean, for a lifetime and infinitely, in theory, basically. Some emerging is that clear to everybody? I mean, maybe a raise of hand would be good for me to understand. Okay.
Sir, what is angiogenesis?
Angiogenesis is, when there is cancer or okay, that would be all. You don't need to raise your hands anymore. So when there are cancer cells, these cancer cells, they grow uncontrollably. Now, when they grow, they are not situated normally where cells are. They are not in the normal architecture. So these cells require blood vessels so that blood supply is required for oxygen, for nutrients, and all that kind of things. So cancer have cells, they have developed these mechanism of angiogenesis. This is essentially, these blood vessels are developed so that it can supply the essential nutrients and oxygen and stuff like that to these cancer cells. Is that clear?
Yes, sir.
Okay. Now the
Sir,
emerging, yeah,
so why
Sir,
are
could
we
you
saying,
sorry.
excuse me, sir, why are we saying evading apoptosis? Is it because apoptosis is beginning in that cycle?
Evading is avoiding, essentially.
Avoiding apoptosis.
Mm. They do not undergo normal, cell death.
Okay, sir.
That makes sense, isn't it?
Yes, sir.
Anything else?
sir, could you again explain the metastasis?
Okay, metastasis is if you know somebody who had cancer, even in case of solid, I mean, tumors or things like that. So what essentially happen is your cancer might occur at a specific location, but then later it travels to some other place, right? So you might have, let's say, a liver cancer, but it might, not remain uh, just localized to the liver. Otherwise, people would have just done a liver transplant and things would have worked fine. It doesn't happen like that. At a later stage of cancer, these cancerous cells, they travel to other tissues, other organs. So there is tumor homing and all. There are various concepts behind it. I'm not going to discuss that. But the thing is, this process is metastasis.
Okay, sir.
Okay, now some emerging hallmarks that were discovered later were deregulating cellular energetics. So essentially, the normal bioenergetics which is involved in a normal cell is different from that of a cancer cell. Let's take an example here. If you know about the glycolysis pathway, about the TCA cycle, you know, glucose to pyruvate, and then there is the TCA cycle. Most of you know about it. Uh,
Yes, sir.
Yes, sir.
ah,
Yes, sir.
so So in case of cancer cells, they go, let's say, through lactic acid, metabolism, lactic acid pathway basically. So pyruvate to lactate is produced. So that will naturally lactate is reduce the pH of the cell, and that's why cancer cells there is a reduced pH essentially. So there is a change in the cellular metabolism, cellular energetics in case of cancer cells. Now another important hallmark is to avoid immune destruction. So what does that mean? So your immune system is very well equipped to recognize any abnormal cell, any cell that is misbehaving, right? It is not normal. So these cancer cells, they have developed mechanisms which so that they can prevent getting recognized by the immune cells of the host, right? Then there is tumor promoting inflammation. So usually inflammation happens to destroy cells, right? So these tumor promoting inflammation, what essentially happens, these tumor cells are more adapted to this inflamed state, right? They do not die, but the usual cells, the normal cells die. So what happens essentially, these tumor cells, they grow in number because they are more adapted to this situation. And that's how that is another hallmark. And then there is genomic instability and mutation, definitely, that we all know that almost, I mean, most of the cancers are caused due to mutations. So we know that already. So that is all about the hallmarks of cancer. So this slide only contains about the same information, how the discovery of mutations were there that produce oncogenes with dominant gain of function and tumor suppressor genes with recessive loss of function, right? And evidence suggests that tumorigenesis is a multi-step process. It doesn't happen overnight. It does not like there is one mutation and the next day you will get cancer. It's not like that. It's a multi-step process. And these steps reflect genetic alterations drive progressive transformation of human cells to malignant tumor cells. Okay. So I hope that is clear. And these are the essential, alterations. These are basically the hallmarks that are important to dictate malignant growth. Now, we'll talk about the first hallmark about the self-sufficiency in growth signals. So have you seen the cell cycle? You know about it. Uh, Most of you should know, actually.
Yes, sir.
Okay, so
Yes, sir.
M phase is the mitotic phase or where the cell division happens. Then after this cell division is happening, the cell, if it has to again divide, it will enter the cell cycle. Otherwise, it will go to this G0 phase or the quiescent phase. The cell does not divide anymore. Now, let's say if it gets signal to divide, it will then come back to G1 phase, and then it will go to S phase where synthesis of DNA and other kind of important things which are precursors which are required for cell division, they will be synthesized. Then it will go to G2 phase and then to M phase. This is entirely a cycle. And this entire cycle is very tightly regulated by various pathways. There are checkpoints at each and every junction, so that if there is any error or if the cell it goes bad in some way, then we can just stop it right there and the cell does not divide further. These kind of mechanisms are active, they are present in your body. However, um, what happens in case of cancer cells, they hijack this machinery and they uh, create their own signals. So we'll talk about it, how that happens. So as you can see here, a RAS pathway. So essentially, usually what happens? Just a minute, still people joining in. There is an extracellular signal that will activate the RAS protein, and this RAS protein it will then go and it will, cause transcription, translation, and replication, stuff like that, and cell division will happen normally. But in case of a mutated RAS protein, this RAS protein will always remain in the active form, even in absence of any signal. So in that case, uh, there will be a constant, there will be a constant signal of the cell to the cell to divide, and it will start dividing uncontrollably. That's one of the simplest mechanisms of self-sufficiency in growth signals. So let's see what usually happens. So if you see a cell, there is like this is the membrane, then this is the nuclear membrane. These are the two membranes. So if there are signals here, and then there will be a receptor here, and this receptor will cause more signaling, and then it will go into the nucleus and cause DNA to be replicated, translated, transcribed, all these kind of things. So what can happen? uh, Essentially, there are three things that could happen. First is that the cellular signal can be altered, right? Are you people following me here?
Yes, sir.
Yes, sir.
So the cellular signal
Yes, sir.
could be altered. Otherwise, there could be a change in the receptor, or there could be some change in the cascade. These three things could happen that could cause, change in the signaling. So essentially, this is the strategy which is involved. So alteration of extracellular growth signals could happen, like this happens in platelet derived growth factors as shown here. So platelet derived growth factors, they are altered based on which these are all the pathways that are changed. So these pathways will lead to either cell survival and cell growth, or it might cause angiogenesis, invasion, and metastasis. As I said, metastasis happens due to a mechanism called EMT, that is endothelial to mesenchymal transition. That's what is written. So essentially, what is important here is that if if the signal is changed, then all its downstream elements, whatever signaling cascade is there, it will also have an effect. Okay. Another thing that could happen is the alteration of cellular transducers. Like you can see, about Herceptin receptors, about EGFR, these kind of things. These are important in terms of breast cancers. Endothelial growth factor is EGFR, growth factor receptors essentially. So these cellular transducers, they could also undergo change. There would be more number of these receptors that will lead to a cascade of cell I mean, a cellular signaling that will lead to cell division and progression of into cancer. Also, the intracellular circuits that translate such signals could also change. So essentially, like you can see the SOS RAF pathway here. If there is a change in this circuit, there is circuit in sense, I mean, this is the basis of systems biology. Do you people know about systems biology?
Yes, sir.
Okay, what would you like to tell me what it is?
So, systems biology, I feel that it's somewhat similar to that of systematics, or maybe it might deal with the biological systems. These are two options that I feel actually.
So as you said about biological systems, essentially what happens is your biological system is, it has a lot of connections in terms of pathways, right? So essentially all these pathways are altered. They are changed, the circuits they might change by themselves, and then there is a change in the system, right? So this study in itself is what happens in case of systems biology. So we usually use programs to do that, to understand or to maybe stimulate or simulate what happens in a biological systems. We try to do that in a computational program. So essentially, if these circuits, as if you come from electronics background or even from any engineering background, these circuits change, that lead to change in growth uh, signals, essentially. So how is insensitivity to anti-growth signal happens? So essentially, the cells, usually normally what happens, they get into the G0 phase if they do not have to divide. But there might be signals that will force the cell out of the quiescent phase. They will be forced to replicate. Okay, that could be one thing. Another thing is that they will be, they will permanently get into a proliferate. They will have a proliferative potential, essentially. So they will acquire differentiation associated traits. So what is differentiation? It is a cell will differentiate. You know what is differentiation and maturation? You must have read about it. Any? Okay, I'll consider you have read about it. We have to go a little bit faster here. So most anti-proliferative signals, they are funneled through the retinoblastoma protein. So, and it's relative. So as you can see, a retinoblastoma protein, it has a transcription factor here attached to it. If this retinoblastoma protein in normal cell, in a normal state, it will be hypophosphorylated. It will have no phosphorylation. So that will cause it to bind with the E2F, the transcription factor, and it will not allow transcription to happen. But if this PRB is phosphorylated, in that case, it will release the transcription factor, and the transcription factor will bind to the DNA, and then transcription will occur, right? So later it was discovered that TGF beta, it is another protein that also has a very important role in regulating this PRB pathway. So TGF beta, it activates SMAD, and then there are P21 and P15 proteins involved that prevent the phosphorylation of retinoblastoma protein or P130, and then it stops the cell cycle to progress. This is exactly the mechanism. Now, how could this be disrupted? There could be downregulation of TGF beta. So TGF beta is not produced enough. There could be some mutation in SMAD4 gene, or there could be some deletion in P15 locus, or let's say P21, that will cause it to be non-functional. So any of this will actually affect this entire signaling pathway, and then this inhibition, this means inhibition, essentially. So inhibition will no longer be there, and the cell will permanently get into a cell cycle. It will always be in in constant proliferative state. Now, is there any question?
Sir, can you move on to the previous slide once? Sir, in this in the normal cell growth conditions, P21 is inhibiting this CDK cyclin, for which PRB gets hyperphosphorylated, right? Rather retinoblastoma protein gets hyperphosphorylated.
Hypophosphorylated. It does not get phosphorylated.
It does not get phosphorylated. So, in the normal cell growth conditions.
I mean, see, phosphorylation, it regulates phosphorylation. I would not say it does not. It is not a 0 and 1 thing. So it will regulate the phosphorylation of retinoblastoma protein. So regulation is important because when it requires the cell to get into cell division, then it will phosphorylate. If it does not require, then it will not phosphorylate. But if this becomes non-functional, or if it becomes aberrant, then the phosphorylation state it will no longer be, in control.
Then what is the basic problem which occurs in cancer cells with respect to this system? I couldn't understand that part.
So that's what I said. So essentially like this TGF beta, how this entire thing, it regulates the cell cycle. That you understood?
Yes, sir.
So what could happen? There are three possibilities, like actually many possibilities. We have only discussed about three. So the TGF beta, that could be, not sufficient enough to for this cycle to continue, essentially. So many cells, they will keep on proliferating because there is not enough TGF beta produced. That could be one thing, right?
Yes, sir.
Another thing is that this MAT4 gene, it could be mutated, right? So it will not be functional. So if any of the elements here is not functional, then it doesn't matter even if others are working. Uh, the cell signaling will not happen properly. That could be one reason. Another thing is that this P15 locus, I mean, there are deletions or mutations which render them actually non-functional. So that could happen. Three possibilities here that could lead to the cell getting into an unlimited proliferative cycle.
Uh, Thank you, sir. But one last question: that what is the role of P15 protein or the
See, it's a signal. So what happens in case of signaling? There is not a single protein that is involved, or like two proteins or three proteins. It is a cascade. Right. So what happens is TGF beta, it does something, it will activate SMAT4. SMAT4 will regulate P21 and P15, which will further regulate the, PRB and P130, their state of phosphorylation in this case. And then it will regulate the cell cycle essentially.
Okay, sir. Thank you.
You don't have to remember all of this. Take care. It's not required. I mean, this is, I know it's a little bit, advanced for your stage. It's not required. I'm just giving you examples. You don't have to know all of this. Don't have to remember it essentially. So apoptosis, it happens. Apoptosis is, programmed cell death. So in case of apoptosis machinery, there are sensors and there are effectors. So sensors, they sense that a cell needs to undergo apoptosis, and then there are effectors. These are receptors, which decide, okay, now the cell should undergo cell death. So most apoptotic signals, they converge in mitochondria. Essentially, it happens in cytochrome C. The signals essentially they go into, and then there are BCL family proteins. These proteins have pro-apoptotic or anti-apoptotic functions. So these are the proteins which are involved here. The examples are given. Now, what do you think would be upregulated in a cancer cell? Would anyone would like to answer?
The anti-apoptotic genes may be upregulated.
Yes. That is exactly what happens. For example, in case of follicular lymphoma, BCL2 is upregulated. So you just need to know these concepts. The entire topic is not about cancer biology, but this is exactly how it happens in case of cancer. Why we are studying it, we'll talk about it later. Then we will talk about the hallmarks of aging. So these hallmarks, again, to become a hallmark, it should manifest itself in a normal process of aging. It's experimental aggravation, or if we increase this hallmark, then it will increase, or it will make the process of aging faster. If we reduce these hallmarks, then it should delay the process of aging. That is essentially the idea. So the hallmarks of aging, you could see there are some common hallmarks here, like genomic instability is there. Very similar to that, we have epigenetic modifications that happen in cancer as well. Deregulated nutrient sensing, very similar to deregulated energetics. So there are some common hallmarks, but they are different in a way. So genomic instability in case of aging could lead to the cell not being functional, and it then undergoes, cell death and that kind of process. So that is genomic instability. Then there is telomere attrition. Anyone would like to say what a telomere is? Do you know what a telomere is?
Palindromic repeats of DNA, or rather stretches of DNA covering the ends of a chromosome.
Right. So, do these ends have any coding sequence? Do they code? Do they form mRNA or
No,
protein?
no, no. They are a
No, they
part
do.
of jump DNA.
Yes, so they have repeats usually. So what happens after each replication, if this is the DNA and at the end there are telomeres, at the end of each replication, it is depleted. So slowly, because there is the priming region and then there is the polymerase sits and then it undergoes the polymerization reaction. So this end is usually depleted at the end of each cycle. So slowly, these telomeres are depleted. They do no longer exist. And if the telomeres are depleted, then the normal genes will be affected. They will be depleted later in the replication cycle. So what happens after the telomeres are depleted? Then the cell understands that it is not possible to divide any further without creating damage to the cellular DNA. So essentially, that is again another hallmark of aging. So usually in aged cell, you will see that telomeres are depleted. So what do you expect would happen in cancer cells?
There would be some mutation so that telomerase activity gets upregulated, for
Exactly,
which
exactly,
uh,
exactly.
for which the telomeres do not shorten and hence uncontrolled cell division occurs and
That
the
is
cell sustains.
exactly That is exactly what happens. So, telomerase activity is retained in case of cancer cells. So the telomere is not reduced any further. So this is again, these are the opposite things which happen in a cell. Then there are epigenetic alterations. Epigenetics means there is no change in the genetics, or there is no genetic change, like there is no mutation as such, but there are some other changes that regulate the cell to divide, or let's say replication, transcription, translation, all these kind of things. So the central dogma is essentially regulated, mostly transcription, translation, these things are regulated through changes which are not genetic. So any example of epigenetic modifications which happen usually? You must have read it in molecular biology.
DNA methylation, histone acetylation.
Yes, yes, these kind of things might happen. So again, it's an hallmark of aging. So usually, how epigenetic state of a cell, what we call as epigenetic state is which genes are switched on and which genes are switched off. These epigenetic state changes with age, right? And then there is loss of proteostasis. So proteostasis is the mechanism how the protein is synthesized, it is maintained, and then it is degraded. So in case of many neurodegenerative diseases also, like Alzheimer's or Parkinson's, like in case of Parkinson's, there is accumulation of a protein called alpha-synuclein that happens due to a faulty ubiquitin proteasome, system. So this ubiquitin proteasome system, it basically degrades these proteins, the misfolded proteins or non-functional proteins. So if this degradation mechanism is lost, then definitely there will be an accumulation of the protein and it will cause many diseases. Then there is deregulated nutrient sensing. So the cell cannot sense nutrients properly, and there is not proper uptake of these nutrients that happen in case of aged cell. Then mitochondrial dysfunction. So all of you would know that mitochondria is the powerhouse of the cell. So if there is a dysfunction in mitochondria, then there will not be enough power to fuel the cells up. And that happens in case of aged cells. Then there is cellular senescence, where the cells lose the ability of regeneration. So if there is any cellular damage, if there is any kind of let's say what is it? Like apoptosis is there. There are cells. There is a void of cells. Cells are required, but the cells cannot divide any further. So essentially, that is what happens in case of senescence. Then there is stem cell exhaustion. So anybody who would like to tell about stem cell? Are you people? Maybe someone other than Antik can answer this. Stem cells. Do you know what they are?
So stem cells are regenerative cells. They show regeneration.
Is regeneration only ability in stem cell or it's something else?
They give rise to the other cells of the body.
But through a process of
I'm not sure, sir.
okay. So
Sir, is it totipotency?
yes, yes, yes. The potency of the cell is very important. So it can differentiate into multiple cell types. Essentially, that is what happens. So stem cells, stem cell exhaustion, what it is. So I can explain it to you using an example. So how long does an RBC live, or a lifespan of an RBC? Do you people know?
120 days.
Yeah, somewhere around that, 70 to 140 days. It's like that. Okay, so RBCs are produced. They are then it dies, but the RBC count in your body or in your blood, essentially, it remains. It is maintained. So how is that happening? Can anyone?
It
Hmm?
from the bone marrow, I think the RBCs
Uh,
are produced.
so hematopoiesis, that is the process, right?
Yeah.
So hematopoiesis में क्या होता है? Initially there is an adult hematopoietic stem cell. So these stem cells, they differentiate into various lineages to give. One lineage gives RBCs, the other gives WBCs, all those kind of things happen. So these stem cells are present in the bone marrow, not only in case of hematopoietic cells, but also other cells also. There are few stem cells that remain there. So in theory, these stem cells could, replicate infinitely. Fine. But what happens after an age? This pool of stem cells are lost, right? They are lost. So there are no longer stem cells available to, differentiate into these RBCs, WBCs and stuff like that. And that is why with age, you will see a reduction in the count. Even the immune system, it affects a lot with age, because this is one of the reasons. There are many other reasons behind it. So is that clear to everybody?
Yes, sir.
Okay, then there is altered intercellular communication. We already talked about it in cancer. So like intercellular communication could lead to like growth, uncontrolled growth. It could also lead to, death. So there is an altered intercellular communication in case of aging. So this is a very important paper. I'll be, attaching this paper along with the slides. Now, as I already said, the hallmarks should fulfill these criteria. That is clear to all of you. Now, apart from these known hallmarks, few others have been discovered very recently. So one of them is compromised autophagy. Autophagy is the mechanism how the cell cleans itself. If there is anything that is damaged, autophagosomes, they will, engulf these kind of, let's say damaged proteins, and it will essentially cleave it off. But if autophagy is compromised, then definitely there will be effects due to that. Then something that you might think it comes out of science fiction. It's microbiome disturbance. Can anybody tell what that is? What is a microbiome? We are talking about this essentially.
So
Nobody.
microbiome is the collection of microbes at a given particular point.
Right, right. In
And
your
like
body.
PSA in our body. For like if we consider for the lungs, the number of microbes present in lungs is the microbiome of lungs.
Yeah. Similarly, we have gut microbiome and many other things, right? So this, it has been observed that once you age, even the microbiome, it also undergoes changes. So that's really a very recent finding. It is very interesting also. So it appears that your cellular, your actually body is not only controlled by everything that you own, but also due to some other factors like these microbes, right? So it is very important. Even in case of cancers, also we have studied that there is a disturbance in microbiome. So how will this be important? Can anybody tell me? Anybody can think if we
Sir, can you
know
repeat
that
the question? Can you repeat the question?
if we know that there is microbiome disturbance, there is a change in the microbiome associated with a particular disease, how will this be helpful for us?
We can target the aging of the microbiomes, and that would in turn help with the other, uh, like the overall impact on the body. We can target the smaller areas.
Right, that is somewhat true. Right, you're actually right in one way. The other way we can actually make help, I mean use this, is that we can do studies, we can and detect some, but if there is a particular feature or which is changing, we can check these microbiomes and we can maybe figure out or predict what kind of problem you might be having from these microbiomes. So diagnostics, right, that could also be used. uh, well then there is altered mechanical properties. Definitely, aged cell will have a difference in. Wait, just a minute. I'm getting some. Okay. Okay. So there is an altered mechanical properties means an aged cell, they are more fragile. They have different mechanical, properties than that of a normal cell. Then there is splicing dysregulation. So what is splicing? Anybody?
Removal of removal of
Removal of
centrons,
introns.
introns and joining of genes.
Does that happen to all cells or every cell, every organism?
No,
So
only for
every
eukaryotes.
organism doesn't
In
have
eukaryote.
exons and introns. So
Only in eukaryotes, right?
only eukaryotes.
So if there is a difference in splicing, then definitely it can cause um, aging, properties of aging. We'll discuss about it here only. Then again, inflammation, as it is really linked to cancer, it is linked to aging also. So you must be knowing, if you have aging population in your homes, they will be complaining of back pain, joint pains, these kind of things. This happen due to inflammation, right? So inflammation is another characteristics that happens, and there are many unknown characteristics also, which I would expect that some of you would do your research and find it out. Yes, do you have any question, Shinjan?
So prokaryotes also undergo splicing.
Do you like to tell me how that
Sir,
happens?
the tRNAs, so the non-coding RNAs undergoes the splicing in prokaryotes, like the tRNA.
Okay, okay, fine. I'll have to look into it. Thank you for letting us know.
Thank you. Sir.
But splicing is more characterized in eukaryotes usually. Then genomic instability. So diseases like Werner's syndrome, Bloom syndrome, they have this genomic instability. So usually chromosomal anomalies could happen, like aneuploidy. There could be change in the copy number. Then transcription, translation, these kind of things can happen. The mitochondrial DNA has a high oxidative environment, and it lacks histones, right? So that is one of the reasons why mitochondrial DNA, they undergo more damage than your normal chromosomal DNA. So that is there, and there are more errors which happen there. And then these are all basically the genomic instability, how that happens. Now, even the nuclear architecture, that also changes due to genomic instability. So as you can see here on the right hand side, this is the the structure of lamins. So these lamins, lamin A, lamin C, lamin B, these are all present inside the nuclear membrane. This is essentially lamins. So these lamins, what they do, they maintain the architecture of the cell. So if there is a nuclear, membrane, it will remain like this and not like this. That happens due to lamins, right? So these lamins are produced through prelamins. They undergo farnesylation at the CAX motif. Farnesylation is a post-translational modification. Now, due to this farnesylation, there is cleavage of this farnesylated moiety by zinc metalloproteases. Proteases cleave proteins, as simple as that, and then there is production of a mature protein. But in case of some diseases, like we will discuss about it in detail. If you know about HGPS, Huntington's Gilford progeria syndrome, what happens is that in this prelamin has a 50 amino acid deletion. So this part is essentially deleted. So there is no CAX motif, there is no farnesylation. I mean, there is farnesylation, but there is no cleavage by zinc metalloprotease. So essentially the lamins, they do not bind here, and the this nucleus, it turns into a blebed structure like this, not like this. So that is what happens. So we'll study in detail about it. So nuclear architecture also changes due to genomic instability. And what else? What other thing can change? So you know about heterochromatin and euchromatin. I would assume that you know about
Yes.
it. Huh.
Yes,
So
sir.
what happens? Heterochromatins, they are closer to the lamins, right? They're closer to the lamin. But once, let's say this lamin is lost and there is no lamin there, so this chromatin will get into its less condensed form, into a relaxed form. So that will cause transcription, right? So this will cause, this will change the entire epigenetic state of the cell. So there are a lot many things which are, linked together. So I hope I can like just make you understand some of it. Let's go to the next slide. So telomere attrition. So essentially there is a as we have already said, a normal cell has a limited replicative capacity to, produce, telomerases and also replicate telomere. Take a minute. Telomerase, replicate telomeres. But in case of, let's say, cancer cells, this telomerase is upregulated. We know that, right? Now, in normal cells, there is absence of telomerase. So if telomerase is not present, then what would happen? The telomeres will no longer work. I mean, sorry, what did I say? Okay, telomeres will no longer be, again, they will not, I mean, the telomeres will always, they will reduce. So in case of telomeres, there is shelter in a complex of proteins that, they make, this a nucleoprotein complex, as you can see here. They make telomeres invisible to DNA repair machinery. So what happens? They will actually they will hold these telomeres. DNA repair mechanism, they cannot, identify these telomeres, and these telomeres will essentially be degraded later. So if there is an increase in shelterin, what would we expect? I think nobody understood this. Am I
Sir,
audible?
will you repeat this once?
So shelterin is a nucleoprotein complex. So there is like it binds to the telomeres. So if this is, let's say, the telomeres, the end of it, and then there is a nucleoprotein complex that binds to it, it will prevent the DNA damage, machinery, DNA repair machinery to actually recognize this telomere. So what happens? The damage that happens to the telomere is no longer repaired. So there is more damage to telomere, there is more loss of telomere. So if the shelterings are increased in a cell, what do you expect? What would happen?
Then the DNA repair machinery would not be able to recognize it. So the telomere will not be degraded, and hence the
Telomere will be degraded rather.
Will
sorry,
be
yeah, the telomere
degraded
will be
more.
degraded more.
So
Will be
that
degraded
will
more for which
that will cause faster aging or slower aging.
that because
Faster
faster
aging.
aging because
That
it's
exactly
degraded.
that is exactly what happens, right? So there are many mechanisms. We'll talk about it in detail later, but anyway, this is just a very brief introduction. Then there is epigenetic alteration. So some of you already said about histone modifications. There is chromatin remodeling, and then there are secondary structures of RNA, like that they also have some role in epigenetics. So like long non-coding RNA, LNC RNAs, then there is miRNAs. They also prevent mRNA translation, right? Then there is DNA methylation. That could What does DNA methylation do if there is a methylation of DNA in promoter region? It prevents
Add methyl groups to any of the bases.
which base?
Adenine, for example.
Adenine is methylated.
Yes, sir. In any sequence, this adenine gets methylated.
So the most methylated base usually is cytosine. Okay, C is the most methylated base. So that's why you must have heard about CpG islands, right? That happens in promoters, that they are present in promoters. Have you heard about it?
No, sir.
Okay, CpG is essentially these are CGs of in a DNA. So CGs, let's say there are some other nucleotides, then again there will be CG. So like this, there will be a lot of CGs present. So they are called CpG islands. So they are usually present in promoter region. So why is this important? This C is prone to methylation, right? So if these Cs are methylated, then what would happen? What do you expect that would happen to the gene if the promoter is methylated?
The gene gets switched off.
Gene guess?
The gene gets switched off
Yes. So
because
there is
the polymerase
it
cannot
reduce
recognize it.
yes, it reduces the transcription. So essentially that is how CpGs are important. So again, CpGs are important for another thing as well. We'll discuss about it. And as you said, there are I mean there is methylation in adenine also, but that happens usually in RNAs, not in DNA. But again, it is possible. And then there are RNA modifications also that happen. So all these things, these are all epigenetic things. So the the DNA is not changed as such, but there are other factors that regulate the central dogma. So that is it about epigenetic changes. Then we will talk about the challenges in DNA replication machinery. So what is the challenge that a DNA replication machinery has? First, it has to find the errors, and then the next challenge is to repair these errors. So there few questions might be in your mind, that how is the genome scanned? How do we how do these replication machinery they recognized the errors, and how they correct it, and what kind of errors are there, and if there is an extensive damage, then how will these errors will be reverted to a normal state. So usually errors are caused. The major reason is error in DNA replication, right? The other reasons which happen are chemical damage and insertion and deletion due to transposons. Some of you said transposon is jumping gene, so you know it. So the mutation or substitution that happens, one of them is I mean, the error, one of the type of error is like substitution. That could be transition or transversion. So what is the difference? Can anybody tell me?
Transition is change of human mutation of the DNA such that a purine changes to another purine and pyrimidine to
Mhm.
pyrimidine. And transversion is purine changes to pyrimidine, pyrimidine changes to purine.
Right. So which is what I mean. Anyway, that is a question beyond the scope of this course. I will not ask it, but I would have asked rather that what kind of change is more common. Uh, but anyway, transitions are more common than transversions. So anyway, coming to the replication machinery, then there is a event called replication slippage. So have you heard about it? I think this is the first time you'll be hearing about it. Do you, any one of you know about replication slippage? No, I think no. I was not aware of replication slippage in my BTech.
No, sir.
Okay, so what happens is there are like repeats. There are repeats, like in this case, there are CAG repeats, right? So there are CAG repeats. Now, this is like the one of the strands. This is the, where it is synthesizing. This is the template strand. So these CAG repeats, usually it will like it will undergo replication in a normal fashion. But since there are a lot of CAGs here, the replication, the polymerase, I mean, the replication machinery will slip. The daughter strand which is produced, it slips, and yet it will form a complementary, you know, uh, association with its template strand, and the DNA replication continues. Is that clear?
Please explain once more.
See, there are repeats of CAGs, right? Many repeats are present. So what is its complementary? It's GTC. GTC is the complementary, this strand. So there are many CAGs and GTCs, in the plus strand and the minus strand. It's present in both the strands. So what happens is, because there are many repeats, so since the polymerase is, it is, using the same basis, so sometimes it might happen that the polymerase is slipping, it slips here. So what, due to the slipping, there is a loop formation, and this, let's say some of the CAGs are here, right? So this happens, but again, it undergoes like the same CAG pattern because it is complementary to GTC here. Is that clear, or again it's not clear? Why does that not happen in a normal, in other places if there are no repeats? So if there are no repeats, then what will happen? Let's say it's ATGC and something like that, CAT, something like that. So if something like this is there, then even if the replication machinery slips, it cannot then find the complementary, pattern, which, it can like, let's say there are CAGs here, right? There is CAG here, there is CAG here. This can slip and it can, bind to GTC here, right? Is that clear?
Yes, sir.
Yes,
But
sir.
if there are no repeats, if there is no GTC, there is something else here, then this cannot slip and sit here. So slippage is not possible if there are no repeats, right? So replication slippage is one of the phenomenon, one of the ways where there, how there is, an error in replication. And in these disease conditions like adult muscular dystrophy, Fragile X syndrome, and even in Huntington's disease, there is, especially in case of Huntington's disease, it happens in case of CAG repeats only. So we will talk about it. So in case of Huntington's disease, there is a change in Huntington gene, which is located in the short arm of the chromosome 4. Right? So there is the Huntington gene here, and what happens? This Huntington gene has around 36 CAG repeats, right? So due to the mechanism of replication slippage, the number of CAG repeats are increased to to more than 36. It goes beyond 42, and then what happens? Huntington's disease. So there is a polyglutamine, residue, because CAG codes for glutamine. So polyglutamine residue is present in a mutant Huntington protein. So what happens if these errors might go unrectified in a cell? So they are not corrected, and during the next round of replication, so let's say in one strand there is 36 CAG repeats, in another strand there is 42 CAG repeats. Now later when this cell will divide, this will be fine, but the 42 one, even if the replication is correct next time, the CAG repeats will increase even if the replication is right, because the template strand has 42 CAG repeats. So in this way, a mutation is fixed. Is that clear? I hope it is. After 1 hour, I've noticed people really do not, concentrate that much. Anyway, effect of UV radiation. So if there is UV damage to DNA, you must have heard about it. Sometimes there is, formation of thymine dimers. Thymine and thymine, they form dimers in the same DNA strand. This happens due to UV damage. There are production of adducts also, which are given here. So whenever a replication, polymerase, it encounters these adducts, or I mean this thymine dimer or these adducts, then the replication stalls. It stops there, right there, because it doesn't know what to do. So that is one of the mechanisms, one of the damages. Another damage is like gamma radiation. It causes double-stranded breaks. So, or a single substitute, I mean, a single strand break could also be there. Single strand breaks are called NICs. You people might be aware of it. You people had genetic engineering, no, rDNA technology already, or it
No,
is
it
there this
is
semester.
it
Oh,
is there this semester.
Yes, sir, it's there.
okay. So you'll maybe learn about these things there again. So there is basic excision repair, which will, repair the DNA if there is a nick. Then there is homologous recombination or non-homologous end joining. So in non-homologous end joining, if there is a break, simply the ends are joined. So some of it is deleted and then the ends are joined just together. That is non-homologous end joining. In case of homologous recombination, let's say this is one strand, this is, and then there is a homologous strand. So what happens is one of the strands would essentially, use the other strand as the template, and it will repair the damage that has been caused, right? So you know about these mechanisms?
Yes, sir.
Yes, sir.
Okay, so if there is a successful repair, then there will be a normal functioning of the cell. If the repair is non-functional, then there will be cell death, apoptosis or necrosis. And let's say if the cell survives with the damage, then there is a chance of, uh, malignancy or tumor formation, these kind of things. So ionizing radiations are, what do they cause? They ionize the DNA backbone, and they can damage the DNA by reactive oxygen species. This is one of them. Then chemical induced DNA damage. So by hydrolysis, this cytosine can turn into uracil. Okay, this is one of the ways. Another thing, what can happen? 5-methyl cytosine can change to thymine. So 5-methyl cytosine actually happens from cytosine to 5-methyl cytosine, and then it changes to thymine. So can you tell me any specific, you know, I mean, use case for this? We already discussed about CPJ islands, did we, or did we not? I mean, just a while back.
Yes, sir.
So C is cytosine.
Yes, sir.
Cytosine can be methylated, right? Are you all following me here?
Yes,
Yes,
sir.
sir.
Once the cytosine is methylated, it then can go, this reaction is called deamination. Deamination, and then it becomes thymine. So essentially what will happen, let's see here, let's see, we have a CG, AT, CG sequence. So here the cytosines are, two of the cytosines are methylated. Fine, everybody is following this?
Yes, sir.
Okay, now this, there will be deamination reaction here. So what will happen? This cytosine will turn into thymine.
Thymine. Yes.
So next time when there will be replication, what will be the complementary strand to this?
AC.
Then AC, then there will be T, A, C, G. So can you see that there is a mutation which has been fixed? I mean, was this the initial sequence? No.
No.
No, it has changed.
So this is again another mechanism how substitutions happen. Very common mechanism. So usually I'm studying virology. In viruses also, we have seen that the most common substitution is C to T. So that happens. Deamination is one of the ways. There could be other ways also. Usually not, it is not always due to deamination, but it is one of the most predominant changes. So anyway, uh, the next part, what happens, this is what is essentially written in the slide, that methylated CpGs are most vulnerable to mutation. Definitely you know why, because of the deamination reaction. There could be deep urination. So what happens again, hydrolysis of the DNA could cause the nitrogen base to be removed, and there is an A basic site. This is called a basic site. These are all DNA damages. We are talking about that. Then we have base analogs. So these base analogs are like of thymines are 5-bromouracil in its keto form and enol form, right? These are analogs. I mean, they can replace these thymine, right? Now, if there is 5-bromouracil in keto form, it will bind to adenine. So thymine also binds to adenine. That is fine. But if it is present in enol form, it is found, it binds to guanine. So what will happen in case of, let's say, ATCG, in place of T, we have now this 5-bromouracil. So I will just make it as an analog is like this. So it will be ATCG. Now it has a enol form, let's say. So it will then bind to TG, GC. Is it clear how this G happened?
Yes, sir.
Yes, sir.
Okay. So in this way, some substitutions do happen and they are then fixed. Sometimes these are recognized, sometimes they are not recognized by the machinery and they are fixed. So even intercalating agents, intercalating, you know, ethidium bromide, very, very common intercalating agent. You must have done acrostic electrophoresis also. So what does intercalating agent do? It I mean, it comes in between the DNA bases, right? The dyes are intercalated in between the DNA bases.
Yes, sir.
Yes, sir.
You people were told to submit the model, you know, that NKJ sir usually gives about DNA model and all that kind of thing. You people did that?
This time we didn't had a model. We had
No, sir. This
poster
time we didn't.
presentation. We had
Okay.
skit and poster
We had
presentation.
poster presentations.
Okay. He used to give that to us usually. You can
Yes,
ask
sir.
him.
Yes. We have seen
Anyway.
that in the labs.
So anyway, so what happens when intercalating agents, they are present in between these DNA. So the distance between this DNA, like the strands, will increase now. So because it increases, so there will be changes. There could be duplications, or you know, if that distance remains like that, so the DNA machinery will think that this is a damage. This is not due to the intercalating agent. So if something we try to do with that kind of DNA, there will be a problem. Uh, So that is it about this. And then we have alkylating agents like nitrosamines. Um, then there is they also cause C to T transitions. Then we have oxidative damage. Oxidative damage happens due to production of ROS. Do you know what ROS is?
Reactive oxygen species.
Why is it produced?
It is mainly produced during the oxidative phosphorylation,
Right.
or
That happens in the mitochondria, right?
uh, the
So
uh,
it changes this like ROS, it changes the guanine residues, one of the damages, into 8-oxoguanine. It's a modified base. Now this 8-oxoguanine, it then changes. I mean, it binds to thymine instead of cytosine. G usually binds to C, but if there is 8-oxoguanine, then it will bind to thymine. So what will happen if this DNA is replicated later? So GC will remain like this. 8-oxoguanine, it might be repaired. So if there is thymine in the template strand, what will it become?
8-oxo-1.
No. I mean, in a normal replication, 8-oxoguanine will not be produced, right? If there is, there will be adenine, right? So this is a
Yes, sir.
G to A change. G to A change is a transition or a transversion,
Transition.
right? So now I think you people can know why transitions are more common. So I mean, you can understand a little bit. So
Yes, sir.
then we will. I mean, I would ask you all to think a little bit about designing experiments with what you have learned. I'm not sure if I have been very clear with all of you. It was the first class for was the material sufficiently I mean clear? I'll take a feedback maybe.
So uh, I in those pathways, the cellular pathways that were causing cancer, especially the cell divisions. If you had just included the pathways in brief, uh, it would have been a bit helpful to understand. Like suppose I couldn't see the P15, the role, the specific role of P15 in that. Like it might be a cascade effect to that. P15 is activating some other protein, which is then activating some other thing, which is then activating P21, which is then regulating the cell cycle, and all those things. But in if we had those pathways in a little brief, then it would have been a bit easy for us to understand, specifically
I
those
mean,
areas.
I do understand what you are trying to say. I will just stop the recording here for the sake of class. Just wait for a minute.
Okay, sir.
Okay.